So today I will be talking about how we at Nelson Lab can assist companies in the qualification of manufacturing equipment in regards of extractables and leachables. So to give an overview, I'll first shortly touch upon some regulatory requirements on extractables and leachables for manufacturing equipment. Then we will look at the first step in the qualification process, which is the risk assessment. We will go over some examples. We will also look into what kind of information is needed to perform a risk assessment. And then we will look at a case study according to the example that's included in USP 1665. And then after extractables data is obtained, an evaluation of the extractables data is needed, which includes an evaluation of the parameters of the study design, and the analytical evaluation threshold can also help in that. So we'll start with some regulatory requirements shortly. Already a long time, there are different regulatory guidances, both in the US and in Europe, that mention that manufacturing equipment should be evaluated for their impact on the drug product, in regards of the impact that they can have on the safety, the identity, quality, purity of the drug product. So they were all very general guidances that tell you what you should evaluate, but they don't exactly say how you should perform that. So if you have a manufacturing process, and especially the manufacturing processes of biological drug products, they contain a lot of different steps and a lot of plastic materials to be assessed. So if you have to start it, you can easily become dazzled on where to begin. So luckily in the last decade there were some interest groups that worked on that, and the most important ones, or the ones that most elaborately worked on that, are the Bioforum group and the USP. So the Bioforum group published the first standardized extraction protocol in 2014, but then it was a very elaborate protocol, so not everybody was very happy with that. And then the USP came with their first draft guidances, and therefore the Bioforum group revised their best practices guide that was published in 2020. So they also published the best practices guide for leachables in 2017. And then, of course, there are the famous chapters that Dr. Dennis Jenke also talked about already elaborately, the USP 665 and USP 1665, that will become official on the 1st of May 2026. So the first step in the qualification of the manufacturing equipment materials is the risk assessment. And the goal of the risk assessment is really to establish the appropriate testing requirements for the different process materials, and that's based on their risk to result in objectionable levels of leachables in the final drug product that might impact their quality or the safety of the drug product. So there are examples included. There's one example included in the BPOC leachables best practices guide. That includes 5 different risk factors, and for each risk factor there are different risk ratings defined. And each risk factor has also their own weight factor. So by multiplying the risk rating with the weight factor for each risk factor and then adding them up, you get a final risk rating and a final risk for your component. And then also in the USP 1665, an example of a risk evaluation matrix is contained. So that includes a 3-step process First, there are 4 different risk dimensions. So for each different risk dimension, a risk level number is assigned, and that results in a numerical risk sequence that can then be linked to the characterization level. And then there is the option to use different mitigation factors to lower the characterization level. So both examples result in 3 risk categories from low to over medium to high, each with their own testing requirements. The testing requirements are somewhat different between what BPOC suggested versus USP. So in the BPOC, the low category only needs compendial testing, medium category needs compendial and extractables testing, and high category needs both compendial, extractables, and leachables testing. In the USP example, or in the USP standard practice guide, based on the risk category, there are different extraction solvents that need to be implemented and different analytical testing. So let's look at what information is needed before we can go into a risk assessment. There is quite a lot of information that needs to be gathered, and it's usually scattered over different departments. So it's very important that different departments come together and that the information is being gathered to include all those different departments. And in the first step, a product contact list needs to be gathered, or a list of product contact materials needs to be set up. So all the materials that come in direct contact with the process stream need to be listed. And it's very important, of course, to not skip any materials and that the list is complete. A material is also uniquely identified by its supplier and grade, which means that if the material that's made of the same material of composition but is obtained from different suppliers will become a different contact material, as it might result in a different extraction profile. This is also true for different grades that are obtained from different suppliers. And in the product contact list, it's very informative if also the material of construction is included for each product contact material. So in a second step, it is very informative to have a schematic production overview. So a schematic overview where all the steps of the production process are shown, and that for each product contact material that we identified, the material is indicated on the production scheme, so it's clear where they are situated in the production process. The volume of the different tanks that are used can be very helpful later on in the assessment. So volume of bulk tanks, buffer tanks, break tanks, and so on. And then, of course, the composition of the contact solution. So for each contact solution that's present in the process stream, list all the ingredients and their respective concentration and pH values. So then finally, for each material in the production process, a few parameters need to be specified. What is the pretreatment of the material, especially sterilization? What's its maximum contact time, including holding times and pausing times? What is the maximum temperature of contact? And then the contact surface area. And we already touched upon this also yesterday. So the contact surface area that should be taken here in account is really the contact with the process stream, so not the surface area of the entire test item. So now we will look at a case study of a risk assessment that's based on the USP 1665 guideline. So in this case study, we are looking at a compounding and filling line of an ophthalmic drug product. So first we made the product contact material list. So we had a list of 21 process items identified as product contact materials. which were tubings, joints, membranes, and filters. And then on the bottom of this slide, you can also see a schematic overview of the production process, or a simplified one, of course. So we start with a tank that contains WFI. The WFI is supplied to a tank B to produce solution 1, which is then filtered and supplied to tank C to produce solution 2 together with the WFI, which then generates the final drug product. This drug product is filtered, goes into a surge tank, and then is filled into the final container through a filling machine. So for each of the 21 process components, a risk level was assigned. So the different risk dimensions were evaluated: materials of construction and the pretreatment, the leaching propensity of the contact solution, contact duration, and process temperature. And then we looked at different mitigating factors on clinical use and clearance or dilution factor. And in that way, we will categorize all the components in different risk levels to check what is the appropriate testing level. So we start with assigning a numerical risk level for each of the risk dimensions. And the first risk dimension is the material of construction or the pretreatment. So for this case, we didn't have really detailed information on the materials of composition, but all the materials were sterilized by autoclaving prior to use, and therefore they were assigned to a high risk level or a risk level 3 for all components. So for the leaching propensity of the contact solution, there is an example table also in USP 1665. So based on the organic content, a risk level can be assigned. Obviously, for the water for injection, it doesn't contain any organic content, so that one is a low risk level or a risk level 1. Solutions 1 and 2 and the final drug product contain solubilizing ingredients above 0.5%. So we categorized them as a high risk level because those components had surfactant-like effects. Then the contact duration ranged from half an hour over 7 days until 13 days of contact. So the 3 risk levels were represented in the manufacturing process. Then the process temperature was 25 degrees, 40 degrees, and 85 degrees. So there we had a risk level 2 for ambient temperature and a risk level 3 for the elevated temperatures. So by assigning these risk levels to each of the risk dimensions, we could come up with a numerical risk sequence that we then could link to a characterization level based on what's included in USP 1665. And in this slide, 4 example test items are listed, and they have a characterization level. One has a characterization level B, or a moderate risk, and the other 3 have a high risk categorization. So now we will look at some mitigation factors to check whether we can bring down the risk of these components. So we were talking here about the topical ophthalmic drug product, which was in a liquid state. So there was no mitigation factor that we can apply for the dosage form. The duration was also chronically dosed, so no mitigation factor for that. But the daily dose volume administered to the patient was only 64 microliters. So based on this very low daily dose volume, we applied a mitigation factor of 1 for clinical use. Then the second mitigation factor was on clearance and dilution. Because we're here in a compounding filling process, there is not really a clearance step in the process that could clear the leachables from the process stream. But we did evaluate a dilution factor that was based on the ratio of the contact surface area of the material versus the minimum homogeneous volume after contact with the test item. So for instance, for tank C, there is a total volume of 1,000 liters. And then we can suppose that the leachables of materials that are before that tank are diluted in the volume of 1,000 liters. And then we applied a mitigation factor if that dilution factor was below 0.001 square meters per liter, which is a limit that's also present in the BPOG example of the risk assessment. So this slide shows a few examples of the calculation of that dilution ratio. So you can see again the schematic overview of the process and the volumes of the different tanks indicated on that. So for the test item 1, which is situated before tank C, the surface area gets diluted, or the leachables get diluted in a high volume in tank C. So the dilution factor is very low, and we can implement a mitigation factor. Then there are 2 test items here in this example, which are situated between the tank C and the surge tank. Test item 2 has a lot of different, a high number of different test items that are present there. So its surface area is higher, and there the dilution ratio is above the limit, and we cannot implement a mitigation factor. And if we look then at the 4th test item, which is situated in the filling machine, there the leachables end up directly into the vial. So the minimum homogeneous volume after contact is 1 vial of 2.5 milliliters. And so the dilution ratio is also quite high, and we do not implement a mitigation factor. So by implementing these mitigation factors, we can bring down the characterization levels. And for the 4 test items that are here in the example, we end up with 2 test items that are characterization level A or a low risk, and 2 test items that have a moderate risk. And those could then be linked to the appropriate testing as outlined in USP 665, which is for the low characterization level of 50% ethanol extract, followed by non-volatile residue determination and UV absorbance. And for the moderate risk level, 50% ethanol extract, which requires a full organic extraction profile. So we started with 21 different process items, and of those, 11 were assigned to risk level A, which needs the minimum testing, and then the other 10 test items, or the other 10 process items, were categorized as level B or medium risk. So now we have determined our testing level that should be applied on the different process items. So extractables data can be gathered and evaluated. So extraction can be performed according to the standardized testing protocols as included, for instance, in the USP. That covers the majority of the pharmaceutical drug products, but not all of them. But it allows an easy comparison also of extraction data of different suppliers, which comes in handy if you have to do material selection. In some cases, there are extractables or some data available from the supplier, but when evaluating these data, it's always important to look whether the study design does cover your intended application. And there are a number of parameters that can be checked there. First of all, of course, it's important that the test item that is used in the extraction study is representative for the test item that you're going to use in your process, in your production process. The pretreatment steps that were performed on the test item in the extraction study should be representative for the pretreatment steps, or at least worst case for the pretreatment steps that are performed on your process item. The composition of the extraction solvents should be worst case in organic content, pH, and polarity. The extraction conditions, time and temperature should be worst case, and the analytical technique should be in line with what you defined with in your risk assessment in degree of testing. So in the end, it is the responsibility of the end user to qualify the process component and show that it's suitable for use in their end application. So now I will go over some examples of evaluation of extractables data that we did in terms of the study design parameters. So in the first example, we evaluated the polypropylene adapter for a tubing, so to connect a tubing. In the real process, we used the polypropylene adapter of 3/8-inch internal diameter that was gamma irradiated on 25 to 40 kilograms. It was in contact with a solution that contained 1.5% drug substance in 0.01% surfactant in a buffer of pH 7.2. And it had transient contact at ambient conditions, and the material was ranked as a medium risk level. So if we looked at the extraction data, the test item that was used for the extraction study was a polypropylene adapter of 3/4-inch internal diameter. And this was evaluated as being suitable as the data could be extrapolated to the process item. So the material was also gamma irradiated at the same dose. So the pretreatment was also representative. But then the extraction was only performed with ultrapure water. And as you can see, the actual contact solution contains some organic content. So this is not representing the actual contact solution. Then the contact conditions in the extraction study were 24 hours at 70 degrees. So that is representative for the transient use at ambient temperature. But the testing that was performed was only USP 661 testing. So there was just a non-volatile residue determined, which is not appropriate for a medium risk level material. So in this example, we identified two gaps on the composition of the extraction solvent and on the analytical techniques. So the conclusion is that there still needs to be extractables data generated for this material. So another example is a Teflon gasket, which is rinsed with water and 70% isopropanol. Before use. It was in contact with a solution that contained an active ingredient in 90% triglycerides and 10% polyethylene glycol. It was in contact with that process stream for 8 hours at 58 degrees, and the material was ranked as a low risk level. So in the extraction data, the same Teflon gasket was used. There was no pre-treatment of the material prior to the extraction study, but we expect that a rinsing procedure would rinse away leachables or extractables of the material, so this could be regarded as a worst case. Then the extraction solvents that were applied were according to the USP standardized extraction protocol. So a low pH solution, a high pH solution, and 50% ethanol. However, if we look at the contact solution, it is a fully organic product. So the standardized extraction protocol would not cover this application. Also, the contact conditions in the extraction study were according to the standardized protocol for 24 hours at 40 degrees, but the real contact solution is at elevated temperature of 58 degrees, so this is also not covered. And then the analytical techniques that were applied were non-volatile residue and UV absorbance in accordance with the low risk level. So in conclusion, we identified here 2 gaps on the composition of the extraction solutions and on the contact conditions. So for this material or for this application that falls outside the scope of the standardized protocol, additional extractables data would need to be generated. And then the last example is a bag system that was used for buffer storage. The bag system contains a film and different port connections and is irradiated before use. It contains a sugar alcohol, or it's used for the storage of a sugar alcohol in phosphate buffer at pH 7. And it can be stored in that bag system for 24 months at room temperature. And it is categorized as a high risk level material. So in the extraction data, the same film and port connections were used for the bag system. So the bag system was seen as representative for the real process material. It was gamma irradiated at a higher dose, which was considered to be worst case for the real irradiation. There were a lot of different extraction solvents used in the extraction study. So the ethanol would cover the organic content of the contact solution, and there are pH extreme solutions that cover the pH of the contact solution. Then there are the contact conditions. So there was an extraction for 112 days at 40 degrees. Now the question is, how do we correlate that with the total shelf life? So one possibility is to use the Arrhenius equation to calculate accelerated aging testing, and what that would represent for 24 months at room temperature. So that are 730 days at 25 degrees. And this would correspond to 258 days at 40 degrees. So if we use that equation, then the contact conditions would not be fully covered. Then the analytical techniques that were applied was a full organics profile that was determined. So that was appropriate for the high risk level. So in this case, it was decided with the customer to do an additional screening leachable study to cover that end of shelf life period. After we have determined that the study design of the extraction study is appropriate for its intended use, we still need to evaluate whether the material is safe for use in the end application. One tool that can be used for evaluation is the analytical evaluation threshold. This threshold defines that compounds that are below that threshold would be safe from a toxicological point of view. The analytical evaluation thresholds can be calculated based on different threshold levels, which I will shortly touch upon in the next slides, that differ over the administration route of the final product. And the calculation of the analytical evaluation threshold also involves some process-specific parameters. So again, here, information on the production process is needed. So in 2006, you probably know that the PQRI published their best practices guide for orally inhaled and nasal drug products. There, 2 different thresholds were defined. The safety concern threshold of 0.15 micrograms per day for compounds that do not trigger a structural activity relationship concern, and the qualification threshold of 5 micrograms per day that triggers for compounds that do not trigger structural activity relationship concerns. Then in 2022, there was also best practices guide for parenteral drug products. There, the safety concern threshold was put at 1.5 micrograms per day for mutagenic impurities. and 5 micrograms per day was the qualification threshold for sensitizers and irritants. And the level of 1.5 micrograms per day might look familiar, as it is similar to the TTC that was put forward in ICHM 7. So ICHM 7 is limited to the mutagenic impurities, and it defines an additional cancer risk of 1 in 100,000 over a lifetime exposure. And it gives the option to raise the TTC level based on the therapy duration. So for the chronically dosed applications, the limit is set at 1.5 micrograms per day. And for non-chronically dosed administrations, the TTC level can be raised. However, this guideline is only limited to mutagenic impurities, and for sensitizers and irritants, no staged approach can be applied, as those compounds have an immediate effect. So in the end, all the compounds that would exceed 5 micrograms per day would need to be evaluated still for irritation and sensitization. So at the end of last year, there was also a publication of the FDA, a draft guidance on topical ophthalmic drug products. There, the thresholds that are mentioned in that document are not dose-based, but concentration-based. And they are defined, 3 different thresholds are defined: a reporting threshold of 1 ppm, an identification threshold of 10 ppm, and a qualification threshold of 20 ppm. So now we will look at how the AAT can be calculated based on the safety concern thresholds. So if we want to calculate an AAT in micrograms per square centimeter, we can divide the safety concern threshold by the maximum daily dose. Then we multiply by the minimum homogeneous volume after contact with the test item, and divide by the maximum contact surface area. Then, in extractable studies, usually screening analysis are used. So the results that are obtained are semi-quantitative or estimated. And so they have some analytical uncertainty still. And therefore, we need to correct the analytical evaluation threshold by dividing it with an analytical uncertainty factor to obtain a final AAT level. And then the compounds that are below the final AAT level should not be further assessed, while compounds that are above the AAT level should be further assessed. And this can be either by toxicological assessment or leachable study. Now, in this equation, we have this minimum volume or minimum homogeneous volume after contact with a process item. So for materials that are used before a bulk tank that contains the whole batch volume of a production process, we might estimate that or suppose that the leachables are homogeneously distributed over that whole batch volume. And indeed, batch volumes are generally quite high. So if we multiply with that number, we arrive at quite high analytical evaluation thresholds. So there will not be a lot of compounds above that high AAT. When materials are used at the very end of the production process in the filling line, the leachables will end up immediately in the final container. And there, the distribution of leachables over the batch volume might not be homogeneous. But in fact, we do not have data on how this distribution is over the batch volume. A worst case assumption can be made that everything would end up in one dose or in one container. But this is a very worst case estimation that will probably not match the reality. But to show the impact of that, I will show an example of a calculation of an analytical evaluation threshold. So in this case, we have a braided silicone tubing that is used for a parenteral drug product with a maximum daily dose of 50 milliliters. And the surface area of the tubing that is in contact with the process stream is 60.9 square centimeters. So in the first case, this braided silicone tubing is situated prior to a pooling tank that contains a volume of 40 liters. So we can assume that all the leachables of that silicone tubing will end up in the 40 liters. So we use the 40 liters to calculate the AET, and we arrive at an AET of 19 micrograms per square centimeter. Then we have to implement still the analytical uncertainty factors. So at Nelson Labs, we have an analytical uncertainty factor of 2 for headspace GCMS and GCMS, which brings us to a final AET of 9.5 micrograms per square centimeter. And for LCMS, we have an analytical uncertainty factor of 5, which brings us to 3.8 micrograms per square centimeter. If we then take the same braided silicone tubing, but it is situated at the very end of the production process prior to filling the vial, then the AET is calculated with, in a worst case assumption that everything would end up in one dose. We calculate the AET with a volume of 1 dose. We arrive at final AET levels of 0.012 micrograms per square centimeter and 0.0048 micrograms per square centimeter. So if we look at the difference, that's a differential factor of 800, which is logical because a pooling tank contains 800 doses. But it's, of course, a huge difference and will also make a huge difference on the evaluation of the results. So if we look at the compounds that were detected in the extraction study, there were 3 compounds in headspace GCMS and 1 in LCMS. So for the pooling tank, if we take the AET that we calculated there, all the compounds fall below the AET. So we could conclude that the tubing is safe for use prior to the pooling tank. But if we look at the worst case calculation of the AET at the end of the production process, by assuming that everything ends up in one dose, then all the components are above the AET. And we would need to do a further assessment, either by a tox assessment or by performing a subsequent leachable study. So that brings us to the conclusion of the presentation. So the qualification of process materials will take more than only analytical testing. The first step is the risk assessment of the product contact materials to establish the appropriate testing level that needs to be applied. Then the extraction data needs to be evaluated, whether the study design is appropriate for the intended use. And the analytical evaluation threshold can be used as a first tool to evaluate whether further assessments are needed. And these further steps can be a toxicological assessment or a leachable study, which will be further discussed by my colleagues later this day. So with that, I would like to thank you for your attention. And please continue.